Hello, everyone. I am again very happy as we have another wonderful occasion of online biological sciences seminar series of TÜBİTAK Research Institute for Fundamental Sciences, which were organized for national and international audiences, relying on the unifying feature of science in a unique intellectual space and trying to minimize the negative impacts of COVID-19 situation in the world on scientific thought and its creation. With this in mind, it is my distinct pleasure to welcome all of you joining us through the Zoom platform, as well as watching us through all social media platforms of TÜBİTAK, the Scientific and Technological Research Council of Turkey. Now, it is also my great pleasure and honor to introduce you our tonight's speaker, Professor Eugene Quinion, world-renowned biologist, a world star of virology from National Center for Biotechnology Information, National Library of Medicine, and National Institutes of Health of United States. He's going to give a great talk about the world of viruses, its global organization and evolution. At the end of the talk, we will have a session for questions, where questions can be asked by sending a message through the chat button of the Zoom platform. Professor Koonin and his research group employ existing and new methods of computational biology to perform research in several major areas, such as empirical, comparative, and evolutionary genomics, deciphering general evolutionary trends, exploitation of genome comparison, classification and evolutionary analysis of protein domains and domains architecture, origin and evolution of viruses. Professor Koonin received his PhD in molecular biology in 1983 from the Department of Biology, Moscow State University. He was a research scientist at the Institute of Poliomyelitis of the Russian Academy of Sciences, and subsequently a senior research scientist and laboratory chief at the Institute of Microbiology of the Russian Academy of Sciences. In 1991, Professor Koonin joined the National Center for Biotechnology Information of United States. He has a number of honors. In particular, in 2018, he received, along with Andrei Linde, Georgy Gamow Prize, and in 2019, he received honor as NIH Distinguished Investigator for his outstanding achievements. Professor Koonin is a member of American Academy of Microbiology, fellow of the American College of Medical Informatics, fellow of the American Academy of Arts and Sciences, a member of National Academy of Sciences of United States, and foreign associate of the European Molecular Biology Organization. At this point, I want to draw your attention to the following words by Professor Eugene Quinion. Professor Quinion says, "Science is not just a job or even career. It is a way of living and thinking. It is effectively a devotion or dedication to creative but rational thinking. These are things that can apply to everything and anything in the world." With this, I want to thank once again Professor Eugene Quinion for joining us for this seminar. We are really looking forward to it. Over to you, Eugene. Good afternoon.
Thank you very much. Good evening, ladies and gentlemen. And thank you very much all for joining the seminar. Thank you very much, Ali Kram, for the too flattering introduction. I will now start sharing my screen. While I'm setting up the presentation, I just want to say that it's such a great pleasure for me and an honor, of course, to participate in this seminar series. Shamefully, I would say, I have never been to Turkey. Although I always wanted to, and it's some consolation. It's a pleasure to visit, at least virtually. So tonight we shall talk about the world of viruses, its global organization and evolution. So as Alikram alluded to in his introduction, I have many areas of research in which I'm actively engaged. But these days, when I'm invited to give a lecture, unless there is a more specific mandate, I usually choose this subject. The world of viruses, its overall architecture and evolution. And there are two reasons for this. Sort of the general ultimate reason and the immediate proximate reason. The immediate reason is very clear. We are living through the completely unprecedented, devastating pandemic of the coronavirus that has dramatically changed our lives and unfortunately has taken many lives over the last 15 months or so. Hopefully, they are much closer to the end of this pandemic than to the beginning, but we are living through this. Since now, virology is at the very forefront of pretty much everyone's thinking in the world, what I'm trying to do is to give a perspective on viruses, to explain, to present a broader perspective, to explain that the world of viruses is diverse, vast, and beautiful in itself, not at all limited to coronavirus or other virus pathogens, but all these find their place in particular parts of that viral universe, as we shall see. This is the approximate reason why I want to talk on this subject. The more fundamental reason is related to this quote from Albert Einstein that I put on this first slide of my lecture. The fact that the world is comprehensible is a miracle, wrote Einstein sometime after he completed the general theory of general relativity. And indeed, it's very striking. There are no miracles per se, but it's very striking when we start realizing that we could, in a certain meaningful sense, understand a large part of the universe surrounding us. Understanding to a significant extent means putting in order, understanding the general organization. And this is, as I will try to show in the next 45 minutes or so, the stage that we have approached in exploring the world of viruses. So I want to share this emergent understanding with you. And the first thing that I want to see, which is important to keep in mind, is that viruses are the dominant entities in our biosphere. Outside the virology, virus ecology, I strongly suspect people do not tend to think that. They would think that the dominant biological entities on the planet are animals, plants, perhaps bacteria for microbiologists, but not viruses. On the other, all those are crucially important. But in a sense, viruses take a special place, because if you just count virus particles in different habitats, say in seawater or in soil or even in the content of our own intestine, you will get astronomical and hyperastronomical numbers. Say in 1 cubic centimeter of seawater, if you go to the to the Mediterranean outside your institute, you will get something between 1 million and 1 billion virus particles, from astronomical to hyperastronomical number, primarily bacteriophages. Typically, although not far from uniformly, there are many more viruses per the same volume in any habitat, 1 to 2 orders of magnitude more than there are cells of any kind. And if we switch to Earth-wide counts, there are at any given moment about 10 to the 31st virus particles. This is literally a hyperastronomical number because there are about 10 to the power 24 stars in the observable part of our universe. So the number of viruses on our planet at any given moment is about 10 million times greater. Something that is so abundant simply cannot be unimportant for understanding the nature, evolution, and ecology of life in general. But there are other reasons to believe viruses are crucially important. First of all, viruses, in addition to their sheer abundance, viruses present the greatest genetic diversity observable on Earth. They present the principal reservoir of novel genes, of genetic novelty. Second of all, or second already, or maybe third or fourth, viruses are the emergence of genetic parasites, such as viruses, is an intrinsic feature or intrinsic trait of evolving life. Wherever you find life, you will find viruses. I will not really go for a theoretical argument in this lecture. It requires a separate one. But we and others have developed a theoretical argument showing that emergence of viruses is inevitable in the course of evolution of life. And it goes hand in hand with empirical argument. That is, whenever any organisms, any group of organisms, any type of cellular life forms is explored in any detail, you will find viruses integrated in the genome or otherwise, but you will find viruses. Then again, this is not all about the fundamental importance of viruses. Viruses show unprecedented diversity of the cycles of replication and expression of genetic information. As first captured by the outstanding virologist and biologist David Baltimore exactly 50 years ago. In all cellular organisms, the strategy of genetic information replication and expression is governed by what is known by central dogma of molecular biology. The flow of genetic information goes from double-stranded DNA, which is our genome, and bacterial genome and archaeal genome, to various kinds of RNA molecules, some of which are structural and function by themselves, and others, the majority, are messenger RNAs that serve as template for translation for protein synthesis. This is a universal of cellular life. These three components, double-stranded DNA, RNA, and protein, and information flowing in this direction, overwhelmingly. Viruses are not quite like that. There is granted, there is a large class of viruses, the largest altogether probably, that follows the same course of information transfer. But there are also, in the world of viruses, also realized all other imaginable ways of genetic information transfer. In particular, there are viruses whose genomes represent not double-stranded DNA, but small single-stranded DNA molecules that are then converted into a double-stranded replicative form, which in turn is used to produce messenger RNA for virus proteins. Then there is something even more unusual from the point of view of cellular organisms, namely RNA viruses. There are all three possible classes, and they're abundant, they're common, as we now know, in the world. And they are all possible classes. The simplest positive sense RNA viruses, whereby mRNA itself is encapsulated into virions and can be used directly in infected cells to produce virus proteins. Negative sense RNA viruses, where it's a complement of the messenger RNA, which is included into virions. And finally, double-stranded RNA viruses, where it's a hybrid of these molecules that is incorporated into the virus particles. And then there are also reverse transcribing viruses that incorporate either positive sense RNA, single-stranded RNA, or double-stranded DNA into their viruses, but are unified by the fact that they use reverse transcription, that is synthesis of DNA on an RNA template in the course of their replication. In other words, crucially, among viruses, they find effectively all imaginable diversity of possible cycles of biological information replication and transfer, suggesting the enticing possibility that viruses actually represent, so to speak, nature's original genetic test systems, the relics of the different types of replication that have been, so to speak, tried by evolution before it focused on the single evolution transmission pathway implemented in all cellular life forms. We shall explore this possibility in today's discussion. Before I go there, I would like to indicate that these Baltimore classes of viruses show a very interesting, non-trivial distribution among different types of hosts. For instance, in bacteria, the great, and in archaea, the great majority of viruses are double-stranded DNA viruses, similar in their replication and expression to cellular life forms, with a significant minority of single-stranded DNA viruses, and effectively almost no RNA viruses, small minority. But eukaryotes are dramatically different in that regard. The majority of viruses in animals, including ourselves, and especially in plants, are actually RNA viruses, mostly positive sense, but also others, as well as reverse transcribing viruses, whereas DNA viruses represent a minority, albeit significant. So this rather striking pattern of the distribution of viruses in the biosphere requires an explanation. We are trying to find it. So let us first discuss the origins of viruses. Undoubtedly, one of the most fundamental deep problems in, I dare say, definitely in evolutionary biology, and I dare say in all of biology. There are three classes of hypotheses that have been considered in the literature for about a century, since first viruses have been explored in any detail in the second decade of the 20th century, and up to this day. To mention first are virus early hypotheses, so to speak, which postulate that viruses have evolved from primordial replicons that existed prior to the advent of the modern type cells some 4 billion years ago. Clearly, this idea, at least in its more modern incarnation, capitalizes on the diversity of the cycles of virus replication and expression that we discussed a minute ago. The second class of hypotheses are the so-called regression hypotheses, or degradation, reductive evolution hypothesis, if you will, which postulate that viruses, the ultimate parasites as they are, have evolved from intracellular parasitic bacteria, gradually being reduced to the virus state. The recent discovery of giant viruses that we are going to discuss towards the end of this lecture added fuel to this type of explanation of virus origin. And finally, last but not least, perhaps the most popular hypothesis on the origin of viruses have to do with the so-called escape genes concept, namely that viruses originate from genes of cellular organisms that somehow have broken with the original host and have become autonomous replicators. For most of the 20th century, the discussion of these possibilities, however interesting and fundamentally important, remain essentially speculative and therefore did not take the central stage in evolutionary biology. This has changed with the massive progress of genomics of viruses and hosts. I should note, maybe I should note it even a minute ago, but I will note now that a major aspect and a major challenge in the study of the evolution of viruses is that there isn't a single gene that would be universally conserved in all kinds of viruses. Much unlike the ribosomal RNA and many components of the translation system and some components of the transcription system that are universally conserved among all cellular organisms from the smallest bacteria, even parasitic bacteria, to mammals and plants. About 100 genes are universally conserved among all cellular organisms. This is patently not the case among viruses. So you cannot, in principle, ever build a single phylogenetic tree of viruses. However, this is one side of the coin. The other side of the coin that gives us leverage to understand, to try and understand the evolution of viruses, is the existence of a certain relatively small number of so-called virus hallmark genes that encode key proteins involved in virus replication and the formation of virion structure. I can schematically show the structures of some of these proteins, the most common ones, on this slide. And these are double jelly roll and single jelly roll. Sorry, excuse me. Double jelly roll and single jelly roll capsid proteins, RNA-dependent RNA polymerases, reverse transcriptases, rolling circle replication initiation, and the nucleases, and certain variety of helicases. And I could add about the same number of other hallmark genes that are connectors, that are hubs in the evolutionary, in the network of the relationship between different viruses. connecting specifically these different Baltimore classes, the classes of viruses with different replication expression cycles, and giving us the chance by comparison, by studying the evolutionary provenance of the and connections between these hallmark genes to get insight into the deep early evolution and even origin of viruses. And so we did in the last few years with my colleagues, in particular Mark Krupovich of Institut Pasteur in Paris. We undertook a concerted efforts to look into the deep roots of the proteins that mediate replication and structure formation in different classes of viruses. And so to begin with the replication modules, behind and underneath the remarkable diversity of these replication machineries, we glean extreme uniformity and, in fact, simplicity. In the sense that the principal replication enzymes of all viruses are all built around the same simple protein fold. The so-called RRM or RNA Recognition Motif domain, which is schematically shown right here. This is a very simple, basic arrangement of these 4 beta strands decorated with these 2 alpha helices, sometimes with additional embellishments, of course, but this is the core structure that is in the heart of all principal enzymes that are involved in the initiation and elongation of the replication of virus genomes, both RNA and DNA. This is a very deep evolutionary roots. This is one of the most universal nucleic acid binding domains on Earth, which gives us some confidence in that the roots of virus replication go to the primordial RNA world, where, as it seems overwhelmingly likely, this domain emerged originally as a cofactor of ribozyme RNA polymerases and subsequently evolved various kinds of enzymatic greatly facilitate and accelerate replication. They then looked in a similar manner into the roots of virus morphogenetic modules, that is the structural proteins. The capsid proteins and other major structural proteins of virions. And here we see a very different picture. What we find is that for the majority of the virus structural proteins, and the list is growing. This is a picture of about 3 years ago, and the list is growing. We identify clear cellular ancestors, suggesting that unlike the central components of the replication machinery, the structural components of viruses have been captured from the host at different stages of evolution, some of these indeed at very early stages. So taken together, these observations lead us to the unifying, so to speak, chimeric scenario of virus origin, whereby viruses have evolved on a certain number of independent occasions, and maybe a couple dozen independent occasions in the history of life, as a result of primordial replicators recruiting major virion structural components from their host. So this concept of the virus origin, which I strongly believe is now best compatible with all the available data, is in a sense a hybrid between virus early and escape genes concepts of virus origin and evolution, those that I showed a few slides ago. So I think we now have this kind of good window revealing the complex origin of viruses during the evolution of life, starting the primordial RNA world, but including also much later events. Okay. So this was our glimpse into the deep past, into the origin and the earliest evolution of viruses. And now we will go back to the present and talk for a moment about more practical matters. As I mentioned in the beginning, we now have a much better grasp of the world of viruses. As it happens almost all the time, at least in much of science, at least, and especially in biology, this progress has been driven by accumulation of empirical data. In particular, by accumulation of virus genomic sequences, which in the last few years has been dramatically advanced by metaviromics. That is by sequencing virus genomes directly from the environment, bypassing the limiting stage of growing viruses on a host in the laboratory, and thereby giving us a chance to look at unbiased, to look at the diversity of viruses that actually exist on this planet in different parts of this planet in an unbiased manner. The reality of it is that for the last few years, a substantial majority and growing majority of new viruses are being discovered, not by traditional methods of that involve, that are painstaking, that involve isolation of viruses from a host, followed by adaptation of viruses to a laboratory host or cell culture, growing, isolating viruses, etc., etc. But rather by direct sequencing in the of virus genome from the environment, metaviromics. So prominent and dominant metaviromics have become that the International Committee on Taxonomy of Viruses has actually made a revolutionary step of admitting the discovery of new, officially admitting the discovery of new viruses and new groups of families and higher groups of viruses purely through metaviromic sequencing. This slide will show you why this radical step has been inevitable. This is also a somewhat outdated slide, about 3 years old, which shows the contribution of metaviromics to sequencing to the known diversity of virus genomes. So on the horizontal axis here, we have a prevalence of different viruses in different locations in the world ocean. This is data from the Tara ocean metagenomic expedition. So in how many of those stations in the ocean the given genome has been detected, and this is a log of abundance. And so you can see these colored circles here correspond to viruses that have been previously isolated by traditional means, viruses with known hosts. And the gray circles correspond to those viruses that that are known only from metavirons. You can see with much clarity that the gray circles dominate, the metaviromic sequences dominate. Moreover, the most abundant ones and the most frequent, the most common ones, all come from metaviromics. Remember, this is logarithmic scale. This is very significant. And of course, if I remake this figure based on today's data, this will be a far more dramatic picture. The present reality is that metaviromics actually dominates the study of virus diversity. And over the next several minutes, we shall explore the expansion of different parts of the virus world brought about primarily by metaviromics. Let's start with RNA viruses. So some 3 years ago, my colleagues and I have harnessed the available genomic data, primarily indeed, as discussed, coming from metaviromics to try and construct the comprehensive phylogeny, reconstruct the comprehensive evolutionary history of RNA of the global RNA viral. Here it's very important to note that although there is not a single universal phylogenetic marker among viruses, there is one, fortunately, among all RNA viruses. The RNA-dependent RNA polymerase gene encoding the enzyme responsible for the replication of virus RNA genome and its transcription to produce messenger RNAs is universally conserved among RNA viruses and serves as the, so to speak, 16th RNA for this part of the virus world. So you can make a single phylogenetic tree, which we try to do. It's given the large number of viral genomes and the extreme diversity, this is a fairly complicated bioinformatic process, procedure which required construction of a dedicated pipeline that is published, that I will not discuss, and that is being constantly refined in our ongoing studies. And by running that pipeline and post-processing the results, we produce a phylogenetic tree of about 5,000 reverse RNA-dependent RNA polymerases rooted by homologous but more distant reverse transcriptases. And here this tree just shown schematically. We will look into the specifics on the next slide. And I just want to indicate the contribution of metaviromic sequences shown in blue here that are here about 50% of the RNA-dependent RNA polymerases, of distinct RNA-dependent RNA polymerases. And when they are redoing this procedure later, year or two later, as we shall see in a few moments, that fraction only increases. So this is the schematic of the resulting universal phylogenetic tree encompassing all the known diversity of RNA viruses. The sequence diversity is high here, and it's difficult to be fully confident in the tree topology, but satisfyingly, we do reveal 5 major branches that are very well supported statistically, and I think are quite solidly established divisions of RNA viruses. 3 of these comprise primarily positive sense RNA viruses with some admixture in the middle of this group of double-stranded RNA viruses. The 4th consists solely of double-stranded RNA viruses, and the 5th includes negative sense RNA viruses that grow from the middle of the double-stranded RNA virus group. According to this tree, and I think this can be considered firmly established, the positive sense RNA viruses, both the simplest replication expression cycle came first, and they came first in prokaryotes, because this deepest group includes this family Leviviridae, which are RNA bacteriophages, such as the famous Q beta and MS2, from which all the enormous diversity of RNA viruses of eukaryotes have evolved. Whereas double-stranded RNA viruses evolved on two independent occasions from the midst of positive sense RNA viruses and negative sense RNA viruses, apparently evolved from the midst of double-strand RNA viruses. Of course, we live now at the times of coronavirus, whether they like it or not. I could not refrain from indicating its position in this tree, and it sits, this group Nidovirales is the order of viruses in which the Coronaviridae is one of the families, and the beta coronaviruses, where the unfortunate SARS-CoV-2 begins, is just one branch within the Coronaviridae family. As you see, it sits deep among other diverse RNA viruses, including very well-known, such as picornaviruses and Kelly viruses, polio, noroviruses, and others, as well as less well-characterized viruses infecting all kinds of hosts, animals, all kinds of eukaryotic hosts, animals, plants, and protists. And I also mapped here the position of other important, some of the other important human pathogens among RNA viruses, such as hepatitis C viruses, the discovery of which was marked by the Nobel Prize in Physiology and Medicine last year, influenza, right here among the negative sense, and Ebola in two branches of negative sense RNA viruses. So this is, I think, a crucial point, that these infamous human pathogens are just branches, small branches, however, whatever their practical importance in the big tree of RNA virus. So basically, I don't have to dwell here because I think I already made these conclusions. I just want to indicate that we seem to be succeeding in establishing a rather clear order in the RNA virus sphere. Once we have the tree is not perhaps even so much important in itself. The tree is crucially important as a scaffold for reconstructing various kinds of evolutionary events that occur in the given part of the biological world, in this case among RNA viruses. And this is what we did by mapping the presence of different proteins and domains in different groups of RNA viruses, and then performing reconstruction using the appropriate statistical methods. What we see here is a trend towards genome complexification. That is, viruses in each of these branches originate as very simple forms, pretty much encoding only the RNA-dependent RNA polymerase and the single capsid protein, and then capturing various kinds of domains, in particular, most interestingly, independently, capturing RNA helicases of different families that apparently enable replication of large RNA genomes. Large only means in this case something like greater than 6 kilobases, but that's large by the standards of the world of RNA viruses. And remarkably, the pinnacle of this complexification trend are none other than the coronaviruses. These are the most complex known RNA genomes that reach the gigantic for RNA viruses size of a little over 40 kilobases through the accretion of the great variety of different domains, many of which, as we now are learning, play important role in pathogenesis of virus diseases, coronavirus disease. All right. I don't have all that much time now, and there are still many things that I want to tell you. So very quickly, in another project that I will very briefly describe with a group of colleagues, we explored a single, albeit large, RNA metavirome that was characterized from a single site in China, in the estuary of the Yangtze River. And it was just a large sample of the very rich, opaque water from that habitat. The result of the analysis of this metagenome was completely striking, in that by exploring this single metagenome, we managed to double the known diversity of RNA viruses, moving from about 5,000 distinct clusters of viruses to more than 10,000, indicating that the diversity, the plentitude, the diversity of RNA viruses that remains to be explored in the biosphere is enormous. They are not even at the level of clusters of relatively closely related viruses, such as in the current classification of virus genera. We cannot even think of saturation just yet. But the other side of the coin, that basically all and each of these newly detected viruses fall into one of these 5 major branches in the tree, indicating, in the previously constructed tree, indicating that at this higher level of the hierarchy, we probably already know the main divisions of RNA viruses. Of course, that does not mean that there aren't really interesting and novel things to discover, novel viruses to discover. And we did indeed discover such interesting and novel viruses with very specific rearrangements in the genomes, in the organizations of RNA-dependent RNA polymerases, in terms of the acquisition of new domains, in various different respects. But all these, nevertheless, fall within the previously established main divisions of RNA viruses. So, not to dwell on this, but I believe we now have a clear-cut scenario for the overall scenario for the evolution of the RNA virus sphere. And now, what I want to do in the remaining short time is to give you a brief tour of the rest of the virus world and the general structure of the virus universe. That will be very brief and perhaps perfunctory, but still gives you some idea, I hope. So the reverse transcriptase virus sphere, retroviruses such as the infamous HIV, hepatinoviruses such as the equally infamous hepatitis B virus, and some less known reverse transcribing viruses that are not found in any prokaryotes, but are virtually universal as a class, as a group among eukaryotes. The main message here is about the same as we had with RNA viruses, namely that they can readily and reliably establish the overall structure of the phylogenetic tree based on the phylogenetic analysis of the reverse transcriptases. The single-strand DNA virusphere, the small viruses that contain single-stranded DNA genomes, usually, but not always, circular and usually, perhaps always, replicating through the so-called rolling circle replication mechanism. Working with Mark Kropovich, we discovered a very interesting route of evolution of these viruses, which appear to have originated on multiple independent occasions from plasmids, not viruses, but plasmids, replicating by these small plasmids, replicating by this rolling circle mechanism through capture of capsid proteins from eukaryotic RNA viruses, the kind of viruses we have been talking about before. So this chimeric scenario of evolution recapitulated on several independent occasions. Most interestingly, these single-stranded DNA viruses also gave rise to small double-stranded DNA viruses, which include important pathogens such as human papilloma. Finally, the double-stranded DNA virusphere, which is more complex than any other part of the virusphere, for which there is no universal marker like the RNA-dependent RNA polymerase in the RNA virusphere. And therefore, we have to turn to different methodologies of evolutionary analysis, such as the study of networks of gene sharing. By exploring such networks where genomes of viruses are connected together through shared homologous genes, we identify using the appropriate methods for network dissection into modules these three major super modules of double-stranded DNA viruses and these two smaller modules. The two super modules include very well familiar tailed bacteriophages such as T4, T7, lambda, etc., which are, surprising or not, related to the animal herpes viruses, which include other very important pathogens. And the second supermodule is the one that unites viruses with the so-called double jelly roll major capsid protein, including such infamous pathogens and smallpox viruses and the related giant viruses. The smaller two modules are the polyoma and papilloma viruses, with which we are already familiar, as well as unusual viruses infecting thermophilic archaea, and these are effectively disconnected from either of the two supermodules. I think we have good ideas already on the roads of evolution of these viruses. Unfortunately, I'm afraid there is no time to discuss this. I wanted to briefly touch upon the giant viruses, such as Mimi viruses and Pandora viruses, which have become quite famous over the last decade or so, and that has been touted as the potential fourth domain of life, possibly originating independently from already extinct form of cells, different from bacteria, eukaryotes, or archaea. What we show by reconstructing the evolution of these viruses is that such evolutionary scenarios, however potentially interesting, are groundless, and that these giant viruses have evolved by capturing multiple genes from the host from much simpler and more typical viruses containing this, that all belong to this double jelly roll capsid protein or supermodule of DNA viruses. The main trend here, like in RNA viruses, but on a different scale, is genomic complexification by capturing genes from. And another point that I like to make in greater detail, but we'll have to make very briefly, is that there are major, despite the consolidation of the overall organization of the world of viruses, and according to the taxonomy of viruses, of which we'll speak in the last couple of minutes of my lecture today, despite that, there are major new groups of viruses that remain to be discovered. And some of these discoveries are totally striking, such as, for instance, the most abundant human-associated viruses are a good example. The most abundant human-associated viruses are not herpes, not papilloma, but are bacteriophages that infect bacteria in our intestine. And in particular, the so-called crustphage-like group of viruses. This crustphage has been discovered 7 years ago purely by methods of metaviromics by Rob Edwards and colleagues in San Diego State University. And then in our work with my collaborators, we greatly, that virus seemed like an orphan, apparently unrelated to any other viruses at the time. With my colleagues, in 2 steps, we have dramatically expanded the family. They have shown that this virus belongs to an expansive family of highly diverse viruses that form a big phylogenetic tree in themselves. Here is the original crustphage, and here are relatives from primarily animal associated, but not necessarily, from all kinds of habitats infecting the bacteria of the phylum Bacteroidetes. And here is yet another expansion that includes some bacteriophages with highly unusual strategies of genome expression. As you see, the difficulty in the presentation of all these findings is that there is too much information that is becoming apparent through analysis of genomes and metagenomes. So to conclude, I want to outline the global architecture and megataxonomy of the virus world. Under the pressure of increasing diversity, the ACTV has formally introduced the highest level of taxonomy, or above kingdom, which does not exist in cellular life forms. It's called officially realm. And there are 4 of these major realms of viruses, each quite large and diverse. Riboviria, which includes both RNA viruses and reverse transcribing viruses, monodnavirus, single-stranded DNA viruses, and 2 realms of double-stranded DNA viruses that differ, that appear to be unrelated, that differ completely in terms of their structural modules, the Rna and Rna Viruses. Together, these 4 realms incorporate the huge majority of all known viruses, but very recently, already this year, 2 additional realms have been formally recognized, one of which is Riboviria, which consists of hepatitis delta, another important human pathogen with very unusual properties related to viroids. In a sense, this is just an infectious RNA, but also encoding structural protein, and something called Rna Viruses, which includes viruses of hyperthermophilic Cranarchiota, characterized very unexpectedly by the presence of A-form DNA in the virions. Crucially, these are extremely interesting realms of viruses, but crucially, they are very small. And my expectation is that the further study and classification of virus diversity will result in many, maybe 10, maybe 20, a certain number of additional small realms like this, but no major ones anymore. These have been discovered. And this I will just have to flip through. We have developed a detailed classification within taxonomy, within each of these realms. Finally, to stop on the global network of evolutionary relationships in the virus world. I think it's most convenient and most informative to show this through three layers of entities and information. One of which includes the conserved domains in virus hallmark proteins, such as the RRM domain that gives rise to all the replication enzymes, various chi, as well as various kinds of capsid proteins. The other level derived from this first one includes these monophyletic realms of viruses. And the third level includes these Baltimore clusters that differ in their strategies of genome replication and expression. What we see here is a complex relationship whereby multiple Baltimore clusters, multiple types of genome replication and expression are brought together within the same monophyletic realm. But conversely, the same Baltimore clusters, such as double-stranded DNA viruses, can be split between as many as 4 and possibly more different monophyletic realms. This is pretty much where I stop. I just want to emphasize once again the crucial importance of metaviromics in the attempts to understand the structure and evolution of the world of viruses. The observation, which is very unexpected to myself. I'm now used to it somewhat, but used to be very unexpected to myself, that we seem to be arriving at a solid and stable overall structure of the virus world. However, of course, a variety of major and most fascinating and practically important details in this overall scheme remain to be filled in. Many quote-unquote mysterious viruses remain to be investigated. And finally, we are actively engaged in deep reconstructions of the ancient viruses, including those of the last common ancestor of eukaryotes and even the last universal cellular ancestor. I think this is where I stop, and it would be unrealistic to acknowledge all lab members and collaborators who have been involved in different aspects of our exploration of the virus world, but I want to mention this group who have been directly involved in this synthesis on the global organization megataxonomy of the virus world that we published last year, and most particularly Mart Kropovich and Valerian Dole, who have been my partners in the study of different aspects of virus evolution over many years, and without whose contributions nothing would have been done. Thank you very much for your attention. I'm more than happy to address any questions that you might have.
Thank you very much, Eugene, for this very deep, comprehensive, intriguing, and exciting talk. Now, we can ask some questions which we received by email. First of all, I want to begin from my questions. Will you please comment on relation between virus population and vaccination rate and mutations? Certainly they are related to each other, and lockdown, of course, related to our present day, since we live in the world of coronavirus.
I sincerely hope that it's coronavirus that exists in our world and that its existence is becoming less comfortable. But yes, all these certainly the mutation rate, mutation rates, mutation balance, and the effective population size effects. Accordingly, the effects of vaccination and lockdowns are all linked in a single population genetic perspective here. Answering these questions full requires a full different lecture. I would be able to give it, but on another occasion. So very briefly, very briefly. Also, clearly, the virus keeps to adapt. We incidentally have a paper coming out in too that gives a catalog of positively selected mutations in the virus genome. It's not all that small. It's not huge. Things have to be kept in perspective. And the primary force that affects the evolution of anything, as a matter of fact, is purifying selection. The selection process eliminates less fit variants. But there is active positive selection adaptation that occurs on tens of sites in the genome, quite many. A minority, but it's significant. Importantly, there are also widespread epistatic interaction between these sites, whereby evolution affects not a single mutation, but combinations. That's about mutation and adaptation. Sometimes it's observed that the evolution of the virus is mostly neutral, and the conclusion is made that there is no adaptation. That's certainly not true. Adaptation continues. One of the key reasons why adaptation keeps going at a sufficiently alarming pace is the fact that, through a combination of circumstances, the virus population in the world has become very large. Accordingly, the standing variation in that population is also very large. And simply by Fisher's fundamental theorem of natural selection, the rate of selection-driven evolution is proportional to the standing variation. And therefore, and standing variation itself is proportional to the effective population size multiplied by the mutation rate. Very basic facts that, however, are often overlooked. Hence, the effect of vaccination, the effects of vaccination, and the effects of lockdown or lack thereof. All these measures lead to the decrease of the effective population size of the virus, and accordingly lower the standing variation and decrease the virus's ability to adapt. Therefore, vaccination is the best. It just cuts it off, and the faster, importantly, the faster it cuts it off, the better, because in partial vaccinated people, and then in unvaccinated, especially in partially vaccinated people or in immunocompromised people that have been vaccinated, there is the most dangerous type. The most dangerous type of selection occurs, namely the selection for escape variants. So far, variants that escape the vaccine. So far, we have not, fortunately, observed escape variants with very high, efficient escape variants with very high, which suggests that these evolutionary pressures to avoid the neutralizing antibodies while retaining the ability to efficiently bind the receptor are not very easily compatible. But nevertheless, and this calls for some degree of optimism, in particular with respect to the efficiency of the vaccination campaign. But not a reason to relax, because we may still see such variants. And what to say? As I said, it's crucial to decrease the effective population size of the virus. And in the at least in the absence of vaccination, these quarantine, reasonable quarantine measures have been very important to achieve that goal. They retain some importance now, but as vaccination proceeds, very fortunately, the contribution of such measures is decreasing.
Okay, thank you.
Not a short answer, but these are important.
Yeah, thank you. Yeah, this is, of course. So we have some questions from participants.
Please.
Do evolutionary mutations display reversibility?
In principle, yes. There is nothing intrinsically irreversible about mutations. In reality, this is rare because there are so many epistatic interactions between different sites in any evolving genome that usually if an evolving organism or evolving virus survives a deleterious mutation, it's not by direct reversal, by suppression that occurs elsewhere.
Thank you. The next question is the following. Did the observed SARS-CoV-2 mutations display correlated patterns?
Well, I have already answered that. Yes, they do. Quite extensive patterns of correlation between different mutations, in particular between some mutations, both for mutations within the spike protein genes, within the nucleoprotein genes, and between those.
Okay, thank you. Within the type 1 envelope viruses, should we worry about future outbreaks from other family members as SARS-CoV-2?
We absolutely and necessarily should. Question is which viruses, and certainly the coronaviruses are a reservoir, but so are flaviviruses. These diseases like dengue and West Nile, etc., tend to be less devastating. They can be terrible on an individual basis, but less devastating epidemiologically because these are vector-transmitted viruses. If a virus comes from the flaviviruses that somehow becomes human-to-human transmittable, that's imminent danger. So far, fortunately, we have not seen many such viruses. And then negative strand RNA viruses, in particular new species and strains of filoviruses related to Ebola and Marburg as well. They are all certainly require watching.
The next question is, what are the keys to preventing and combating future pandemics?
Yeah, I try to describe the broad virus world, but questions are about the pandemic. I understand. Well, and I must say that, you know, epidemiology, we have expanded our work in the last year, evidently. We have done in particular a lot on coronavirus evolution. But I should say that epidemiology generally is not my specialty. That has to be admitted right away. So it's more like, you know, informed common sense things that I can, somewhat informed common sense things that I can say. But there are evidently many lessons, and one is monitoring. It's a big challenge, but I think it's not unsurmountable to monitor the environments with which humans get in touch, in particular, through globalization, deforestation, and the like, and identify candidates and develop methodologies to identify candidates. We are not there, and it's not easy, but in a somewhat longer perspective, it is doable. And that's one key. The other one is evidently this quarantine strategy, and the very simple lesson that if it is when it is done swiftly and decisively, it is far more efficient and eventually far less painful than if you know the drastic measures are introduced later. And the third, this is very obvious, but it's just you know, just a wonderful breakthrough, which the creation of RNA vaccines, whereby whatever appears, whatever is captured, hopefully at an early stage, the vaccine can be created now very, very quickly without turning to the difficult, time-consuming processes and dangerous processes of virus adaptation. So this is a great, great breakthrough, scientific breakthrough, which is, by the way, underlain by important fundamental observations on what makes mRNA translatable and stable once transfected into cells. There are lots of fascinating observations on these requirements. So I think these achievements and these challenges, I cannot, as I say, I cannot go too deep into this, not only because of the lack of time, but also because I'm not an expert in epidemiology.
Last question. Do we know that if other coronavirus had a similar virulence patterns at a certain stage of the evolution as in SARS-CoV-2?
Whether or not we should expect another virus with similar characteristics. I would say, given the diversity of coronaviruses in nature, this is highly likely. Moreover, we cannot rule out the possibility, the scary possibility, that a virus with perhaps less likely but scary possibility that a virus with a high case fatality ratio, such as MERS, but the transmissibility, infectivity features of SARS-CoV-2 emerges at some point, and then we might face truly devastating epidemic, it's less likely, the combination of these properties is less likely, but given the vast reservoir, how can we rule it out? One have to be very watchful of these.
Mm-hmm. I don't see more questions. So
no, it's fairly late in Turkey now.
So if there are no more questions, perhaps we can stop here. What do you think?
I'm delighted to answer questions as long as we keep coming. But if they are not, then thank you all very much.
Thank you very much. It was our pleasure and honor to listen to your lecture seminar today. I hope we will repeat this in many cases in person in future. So
thank you very much, Alitra. Yeah. Very hopefully we repeat this in person, but virtual is also fairly effective, I think.
Yeah.
Much less fun, yeah, but fairly effective.
That's true. That's true.
All right.
So
thank you very much.
Thank you once again.